Thank you all for being here. And that was a great overview to kind of put everything in perspective. So I've got a really difficult task in trying to summarize this scientific conference, which we're still kind of, I think, all sort of thinking about what happened and what we learned from it. It was a lot of information in a short period of time. But I think there's some things that we already know from what we learned in the conference, and I'm going to try to summarize it as best I can, if I can figure out how to operate this thing. I think I'll just use the arrow keys. Okay, that works. So I mentioned last night at the dinner these scientific conferences matter. That sometimes they seem like a lot of trouble, and you don't really know what they're for. But why have a research conference? We've been having these family conferences and registry, research network conferences for 8 years, and this was something different. We added a scientific conference at the beginning. I think because, we all know the patients that we deal with, we all know the issues that we deal with and the problems and the questions we have, but a lot of times in science, the real breakthroughs in research come from outside the field. They come from somebody working on cancer or somebody that's working on lung development or something that is not somebody we interact with on a daily basis. So bringing in new ideas, bringing in people from outside is very important. Science moves really fast, and you have to keep up with the best new ideas and innovations that are coming out there, or you end up getting stuck. You end up working with how to get information on DVDs when you really should be making a smartphone app. If to use an analogy, the technology moves very quickly, the scientific knowledge moves very quickly, and it's important to bring that to the table. Collaborations are critical. I think the days of the scientist working in the basement laboratory and running upstairs, eureka, I found it, that doesn't happen anymore. People don't do that. You can't just go in a lab and discover a cure for a disease. You have to collaborate with people from different disciplines and different fields, different institutions, different countries, and it really takes a big group collaborative effort to make scientific breakthroughs, and so I think that these meetings bring people together, and I think a critical thing is young investigators need to be inspired and they need to be encouraged. It is a daunting thing to become a researcher. You go to medical school, you learn about science, you learn about medicine, you learn how to take care of patients, and you get your training, you learn how to take care of the patients in your area, whether it's obstetrics or pediatrics or whatever, and then you go to fellowship and somebody tells you in 2 years you need to become a researcher. And you're learning oftentimes a whole new discipline you've never learned before, and then you have to go out there and compete for grant funding with people, who have been doing it for 40 years and have millions of dollars of funding and that sort of thing. So it's not an easy thing for these people who are coming out of fellowship or out of a PhD program to start a career in research, and to come to a meeting like this and have somebody who's a famous researcher come up and talk to them about their poster and say, well, this is really great. Did you think about this? Did you think about that? That gives them the intestinal fortitude to go back in the lab and do it again and write another grant proposal and that kind of thing. So I think these conferences really are critical. You guys bring the magic. I think the wonderful thing about this conference is having the families here. And you could see, I tell you, one of the highlights for me, there are many highlights of this conference, but one of them was Heather Mating talked about her experience with Luke and transplant and all those sorts of things. And you could hear a pin drop in that room. I mean, these are busy people. If you go to a meeting with most of the people who were at that conference at work, half of them are on their iPad or cell phone. They're like submitting a grant proposal during the thing. Nobody was looking, everybody was looking at Heather. And then a little bit later, we're having another conference, and Luke walked in the room, and everybody's eyes were on that kid and following him, coming in and stuff. And that, to me, was one of the key moments of the conference. So you guys really are very important in this process, because we need inspiration. We need something to keep us going when things are tough. We need perspective. We need to realize that, we're trying to understand this very arcane question, whether it's a clinical question or a research, basic research question. We need some perspective, what it's all about, and you guys and your kids are what it's all about. We need to be kept honest. Sometimes we follow rabbit trails. We get following a particular set of questions and it leads us to something else and we think it's really important, but sometimes we need to push the reset button and see what we're doing. And the families need to know, I think, that somebody's out there trying to figure it all out. I mean, you're dealing with your child on a day-to-day basis and you're dealing with your anxiety and your fear and that sort of thing. And I think seeing the scientists and seeing what they're doing is important. And I think we have examples from other fields where you get families and researchers together and magic happens. And I think probably the best example of that recently is the LAM Foundation, lymphangioliomyomatosis, which is a lung disease that affects young women. And those women are passionate about research. They get in the scientist's face, they get in the doctor's face, they get a registry going, and they get breakthroughs, and they get a drug that actually, has some clinical benefit in a very short period of time. Now, that's one disease, a very focused effort, and a lot of things came together, and Lisa Young can tell you more details about that story. But I think when you get families that are passionate and researchers that are passionate and clinicians that are passionate together, I think magic happens. So I'm going to try to go over some of the concepts that we learned about during the scientific conference. Some of them are a little bit complicated, even for me, maybe especially for me, but I wanted to give you a flavor for some of the things we talked about and some of the things that may have some benefit down the road. So we learned about this concept of proteostasis. Most of us don't even, after hearing Dr. Balch's talk, don't even really know what that means, but it sort of means keeping proteins in balance. And I think the best analogy he had is, a cell, which is the sort of the fundamental unit of a body, of an organism. A cell is like a city, and it has important things like City Hall, and it has fun things like SeaWorld and that sort of thing, but it also has, garbage. People produce garbage, and if you don't have somebody to go around and take out the garbage, it just builds up and you end up with a mess. I mean, if New York City didn't have garbage in 2 or 3 days, it would be impossible to live there. You've got to have sewer services, you've got to have water, you've got to have electricity, and that's what this whole sort of proteostasis network is, all these maintenance functions that a cell has that keep things in balance. And the concept that we learned is that over, whether you believe it's by design or it evolved, in any case, over time, we have ended up with this very sophisticated system that even in the face of an abnormal protein or a mutated gene or a disease, a severe infection, can find the right balance. And a lot of times the drugs that we use will throw the whole system off balance in order to fix one part. So sometimes understanding kind of the fine-tuning may be able to help us find drugs that just kind of nudge the system a little bit instead of hitting it with a sledgehammer and put things back in balance. So there are people who are theoretical mathematicians and computer geeks and stuff that study all these networks and how things interact and try to understand how the balance happens. And I think there's going to be some real breakthroughs coming from this type of work. It's very difficult, it's very hard to understand, but the CF Foundation has invested in this approach, and I think this idea of cellular balance and proteostasis network is really important. And people like Robin are now looking, in a very practical way, looking at lung washing fluid from kids with different types of child disorders and seeing how the proteins may be unbalanced, or if there are particular proteins in there that can help us figure out how the cells are out of balance and put them back in order. So I think that's an important area of research. We learned about the cloud, and this really has to do with the same thing, but the idea is that cells try to find an energy state, some place where they can achieve an area of balance, and that all these different pathways can affect that energy state. And if one pathway is disturbed, it may not be possible to fix it. Let's say there's a genetic mutation so that you can't go down one pathway. You can find another way to go down. It's sort of like, if you're skiing and your favorite ski run, had an avalanche and is closed, you just find another way to get down the mountain. And so some of the research breakthroughs may come from learning how to push cells down another energy state. Genetics is very important. Aaron gave us an update on what's going on in genetics. I think the key things is that this field moves fast and it never stops moving. And you have to incorporate new technologies and new approaches all the time. I think the ability to now really understand genetic abnormalities in the context of an individual family is more than we would have dreamed about even 3 or 4 years ago. But without excellent clinical information, the genetics really don't mean very much. So we really need that registry data. We really need the context of understanding exactly, what the child's illness is and how it's manifested so that we can understand the genetics. And so I think the registry data is important, and you guys are a big part of that. Epigenetics is a relatively newly understood field, and basically it has to do with the fact that DNA is just a string of molecules, and it acts like an alphabet. And the alphabet, you put the letters together in certain ways, they make certain words, and you put the words together and they make a language. But the whole thing is modified by the way that the DNA is packaged inside the cell, and the way that the DNA is accessible to the machinery inside the cell that reads and decodes the DNA, and that's what we call epigenetics. And the thing that we've learned about epigenetics is two people may have very similar genetics. Let's say they're identical twins, and they grow up in 2 different places, and one of them is exposed to a particular thing in the environment, pollution, radiation, whatever it might be. That's going to alter their DNA so that even though they started off with the same DNA, they may end up with very different problems because of the environment. So the environment changes the genes and how they function, and that information actually can be passed on. Epigenetic information can be passed on from one cell to the next and from a parent to a child. So understanding the system is important. It's an extremely complicated field. There's an incredible revolution in technology and our ability to measure epigenetic changes, but it's extremely expensive. The positive side of this is that there's some therapies that are already available that tweak the epigenetic control system, and some of these are already being used for certain cancers. And they may be useful to modify inflammation, or they may be useful to modify the fibrosis process that's common to many interstitial lung diseases. Animal models. So we learned from Dr. Whitsitt. Dr. Whitsitt at Cincinnati has really moved lung research forward in many ways, largely through taking advantage of the ability to manipulate genes in mice. You may think that making a mouse glow green in fluorescent light is a stupid idea. Why would you want to do that other than the fact that it makes a cute picture? But being able to detect where a cell comes from, where it goes, and what happens to it during development is critical. And sometimes doing things like putting a jellyfish protein that glows in the dark on a lung gene helps us to understand, how the lung works and what goes wrong during disease. So these things are really important. There are other animal models, fruit flies, zebrafish. You may think these have absolutely nothing to do with human beings. Why are we wasting millions of dollars studying them? But sometimes these very critical, fundamental ways to understand how cells operate and how molecules interact leads to big breakthroughs in other fields. So we would like to avoid making animals sick in order to find out scientific discoveries, but we can't experiment on your child and your family members, and we need something that's more than just a a cell in a dish or, you know, some DNA in a test tube to understand, you know, how these diseases work. We need something that's a living organism that has lungs that we can study. And so we need animal models. And most of the major breakthroughs in the past 20 years really have been facilitated by having these things available. Stem cells, a big topic. A lot of people are interested in this, and we had several excellent presentations related to stem cells. I think that there's a lot of hope here. So the basic idea to try to put things in perspective is that right after fertilization, you end up with an embryo which is just a few cells, and those cells have unlimited potential. They all look the same to start off, but some of them are going to end up, you know, creating hair, and some of them are going to end up creating eyes, and some of them are going to end up creating lungs and that sort of thing. So there's a lot of potential for repair and rebuilding damaged organs and tissues from cells that are in that early sort of primitive state where they can go in different ways. Down the road, cells differentiate. They commit themselves to becoming a lung or becoming a kidney or whatever. But what we find is that there are cells even in mature organs in adults that still have the potential to differentiate into another cell type and maybe help fix a problem. We can also create stem cells in order to learn about disease and treat. And this is a really important concept, and this is something I think is worth investing some time and thought in. So we can take a skin cell and through a series of genetic manipulations make it into a stem cell. Why is that important? Why is that important? I'll give you an example from somebody who's working close to here, and then I'll let Dr. Cotton say a few words about this. But we have somebody here who studies autism. There's no way to get brain tissue from a child with autism to study, you know, unless the child's in an accident and dies or something. You know, you can't study autism brain tissue in a dish, so you just have to figure out some way to try to model that. Well, he actually is collecting teeth when kids lose their teeth, and from the pulp of the teeth that come out, he's able to generate stem cells. He's able to turn those stem cells into nerve cells in a dish, and suddenly he's got a way to study nerve cells from kids with autism, which would not have been possible before. So this is the kind of thing we're able to do now with some of the scientific breakthroughs that we have, and it gives us a lot of opportunities both to learn about disease and then to come up with new therapies. We still have a lot to learn in this area. I thought maybe I'd have Dr. Cotton come up as one of our featured speakers, Daryl Cotton from Boston, and he's doing a lot of work in this area of induced pluripotent cells.
Thanks for the opportunity to speak to you. I just have a couple of comments to make. So I'm Daryl Cotton from Boston University, and Laertes Economou in the back will be speaking to you later. You can wave, Laertes. There's Laertes in the back. Yeah, so this afternoon he'll speak to you about the stem cells we've been making from kids with child. We have 6 patients that we've received anonymous samples from around the country, and we've made a stem cell bank funded by your foundation, by the Child Foundation, and those cells will be shared with any academic researcher free of charge across the world who wants to work on better understanding these diseases. So I have a few messages that are important. One is to thank you for supporting us and to tell you the importance of this foundation. So I'm an adult pulmonologist and a stem cell biologist, and I wouldn't normally work on child, right? So at American Thoracic Society, the leaders of your foundation, Robin Dieterding specifically, approached me and said, you do all this incredible stem cell work, and kind of shook me by the shoulders and said, you have to look over here and help these kids. And Robin kind of dragged us in, very willingly, but she dragged us into the community, and we became really obsessed with your children and trying to better understand their problems and help them one day. And so that's what a foundation like this does. You attract people from outside the field to come and try to bring our research that normally is focused on adult diseases like emphysema to working on your kids. And that's really important. That's how a field moves forward. The cells themselves, Laertes will show you photos of later and discuss with you and answer your questions this afternoon. But I wanted to tell you why the grant you gave us, the first Child Foundation grant, was important, because it brought us into the field, and now we're hooked, and we're here to stay. The second message is one of the importance of protecting research subjects with anonymity. So we received 6 patient samples so far we've made into stem cells from kids with child. And we received those from investigators like Robin in Denver and Aaron Hamvas in St. Louis, or Larry Nogi in Johns Hopkins, and Sesh Cole in St. Louis. And we received them as patient 1, patient 3, patient 4. And actually, that's good and bad. So the good part is it's really important these samples are anonymized so that we don't know who they're from. We can do our research objectively and bank them. And it's also important because long after we're all gone, everybody in this room is long and dead, the cells of your kids are going to live on in our cell banks, in the stem cell banks, and they're going to be shared around the world continually. And it's really important that the samples remain anonymous to protect your families and your kids' identities. The bad part is that, of course, then we lose the face of the child. And so when I present our data, patient 3 and patient 4, that as we have to, and you'll see Laertes presenting this to you this afternoon, patient 1 and 4, they're not just numbers to us. We care a lot about your kids, and we come to these meetings because when I see Heather or Anne present and I see the photos of your kids, that's who we think of when we look under the microscope. So I see Luke running around. I know this. I don't think the cells are from him. I can't know that, right? We're protected from knowing that. But still, I have his face in mind when we're doing our work, and that really gives us meaning and motivation. So that's really, really important to us. The last message I want to give you is that the importance of basic science research. So this is I just want to reiterate Jim's comments. So we're going to enter an election year, and you're going to hear a lot of almost condescending comments from various politicians talking about wasting money on fruit flies and worm research, right? When we have suffering children that can't breathe. That seems ridiculous, doesn't it? So we in this room know better, and it is our responsibility, all of us, physicians, and nobody cares about the physicians and the researchers. We're just self-serving people saying fund our work. But the families, the parents of these kids, you guys, actually the politicians and the public do care about what you have to say much more. And so it's your responsibility to educate the public and the politicians, to tell them that there's no such thing as translational or non-translational research. All research is translational. What I mean is, your kids with knee high, we saw a research presentation yesterday with stains from the lung biopsies of your kids, and it turns out that perhaps part of the disease seems to be related to a protein called Notch that's working in the lung airway. Notch is a familiar word to you. It's named because a scientist decades ago working on fruit flies with no goal of clinical translation or helping anyone, just a curiosity to know what made a fruit fly's wing, observed that some of the flies had a notch in their wings. And that led to the discovery of a protein called Notch. And that one day led to a whole biology of how Notch signaling works. And it led to an investigator staining your kid's lungs and finding Notch protein right there at the neuroendocrine bodies. So all research is translational. And it is your responsibility to educate Congress and the public that actually the main discoveries that have improved human health in our society, this has been studied more than half the time, it is based on basic science discoveries that had no goal of clinical translation. And so funding fruit fly and worm and other research, or the researchers that wanted to know what made the jellyfish glow green, that have led to gene therapy discoveries that make the blind see already and will one day help your children's lungs, that those experiments are what helps human health. And so you tell the politicians and the public that when they scoff at fruit fly and worm research, you tell them the lives of your children depend on that research. So thank you very much for your support in bringing us into the community, and especially showing us the faces of your children. Thanks.
I could probably stop there because I think that says it all. I'm going to try to get through a couple more points of things we learned. Neuroendocrine cells, we talked, a couple of talks focused on the role of these cells and where they come from, and some thoughts about what they do in the lung. I think this is very exciting new research. It's amazing to me that we've known that neuroendocrine cells exist for, I don't know how long, probably 60, 70 years. And we still don't know exactly what they do and why they're there. And I think there's going to be something very important that comes out of this. And my prediction is that what we learn about neuroendocrine cells is going to really help us understand a lot of things about how the lung works in many contexts. So, a lot of respect for the researchers that are plugging away in this area. Like we said, the breakthroughs may come from another field. Something new we learned about is exosomes or microparticles or microvesicles. These are little tiny bits of cells that I guess in the past we didn't see them because we didn't have microscopes or instruments that were powerful enough to see them, or we thought they were just little bits of trash that the cells were tossing out. But it turns out that cells release little parts of themselves that go and talk to other cells and can actually change the way the other cells function. And it's turned out that these are very important in certain conditions, such as cancer, immune function, and I thought I was really smart because we started finding that a fibroblast that we study in culture release these exosomes, and we thought this was really going to change the way we think about lung fibrosis. And I think that may be the case, but I found out that there are other scientists who are far ahead of me on this, which is what you find out about these at these meetings, and it's a good thing because it motivates me to go back and speed up the experiments, but also motivates me to collaborate with some of the people who are looking at this in other ways. But I think this is going to be an important area. Dr. Kurimbanis from Boston showed that you could take these little microparticles from a circulating stem cell and give them to mice with impaired lung development as a result of exposure to high oxygen, and that these little microparticles were capable of transmitting information that repaired the lung almost to normal in terms of its growth and development. So these little things are important, and I think there's a lot of new areas. We sometimes think we're stuck in a particular field, and then a breakthrough comes along which really changes the landscape. So I think this is going to be important, whether it's a way of diagnosing an interstitial lung disease by doing a blood or urine sample, or whether it's a possibility for a new type of treatment. Age matters. I think I've been saying this for ever since I've been in research, because I do research on an adult lung disease, idiopathic pulmonary fibrosis, and yet I'm a pediatric pulmonologist. But I think that by sort of crossing over by pediatricians trying to understand what's happening in adult lung diseases, and by people who are adult pulmonologists and researchers trying to understand what goes on in child disorders, we're really going to make the connections of understanding how genetic abnormality, for instance, changes the lung in a newborn versus a 10-year-old versus a 70-year-old. And I think that's where we're going to really get the breakthroughs that allow us to create new therapies. So I want to encourage you to collaborate with the Coalition for Pulmonary Fibrosis, the Pulmonary Fibrosis Foundation, alpha-1 antitrypsin, the other organizations that deal with diseases that are more common in adults, because I think we can all learn from each other. So where do we go from here? In a way, we don't really know. That's the wonderful thing about science. We don't know where that next breakthrough is going to come from. We just keep doing what we're doing and moving forward. But these connections that we build at these meetings often are critical in leading to breakthrough. I think some things we do know is that registry data are critical. So it's very important to keep supporting that in any way that we can. I think awareness is critical, as Dr. Cotton pointed out, not just awareness about child and your child's particular disease, but awareness about scientific research and the importance of basic research. So I think your advocacy is important. Fundraising is critical. Science is very expensive, and the government doesn't have money to fund everybody's research. Young investigators are critical. This is where the new ideas come from. And so your supporting an ATS grant, I think, has been a huge breakthrough. And I think we've started off with a good one, as you'll hear a little bit later. So things we know: research is important. As I said, we don't know where the answer is coming from, but we do have some pretty good ideas, some pretty good leads. You cannot raise enough money for all the needed research. You just can't. Even the CF Foundation, as far down the road as they are and as organized as they are and as wealthy as they are, doesn't have enough money to fund all the research that's needed in CF. So advocacy is really important. Young investigators are important. Technology is important. Some foundations will pour money into supporting a particular technological advancement, and I think that's good, but the answers don't always come from technology. And just think about, your new smartphone. You're the best guy on the block for about a day, and then somebody has a newer one that does something that yours doesn't do. So the same is true of scientific technology. People probably more important to invest in than technology. But support matters, and it comes from a lot of different avenues. So keep up with the fundraising and keep contacting other organizations that may take an interest in child for whatever reason and may provide additional support. I didn't think that the surfers were going to be interested in child, but because the surfers are connected with Ambry and Ambry's interested in genetics, then suddenly we have professional surfers who are teaching your kids to surf, but may also help with fundraising and awareness. So everything's connected. So what you can do, the first thought is what my daughter always says to me when I ask her to do something, which is, really? I mean, I'm 17, I'm trying to apply to college, and you want me to clean the kitchen, You guys are facing a lot. You guys just being here, dealing with your kids on a day-to-day basis, we can't ask you to do much more, but I think it's critical that you do what you can. Obviously, focus on your families, focus on your children, but to the extent that you can, awareness is important. Stay involved with the Public Advisory Roundtable and the ATS. Greg Porta has been a diehard advocate for child in the ATS, and I think it's really made a huge difference. Keep going to the meetings so the scientists can see you and you can see them. Fight for answers for your child's diagnosis. That's important, but remember the cloud. Everything's connected in the cells, and everything's connected in science and research, and the breakthrough for your child's disease may come from cancer research or neurologic research. So